First, the RNA polymerase attaches a promoter sequence and then starts unzipping the DNA double helix by breaking the hydrogen bonds between the two strands. Because the sense strand is the one that needs to be transported, the antisense strand acts as a template for the RNA polymerase as it brings in the free RNA nucleoside triphosphates, which align in complementary base pairs just like DNA. The only difference between DNA and RNA is that RNA doesn't contain thymine, but a closely related substitute called uracil. Two of the phosphates in the RNA nucleoside triphosphates undergo hydrolysis to provide the energy needed for a covalent bond to form between the remaining phosphate and the next nucleoside. Because only exons are wanted in the mature mRNA, an enzyme cuts out the introns between the exons and sticks the now mature mRNA back together. The mRNA now leaves the nucleus via the nuclear pores and travels to an either bound or free ribosome for translation. A tRNA molecule is recognized by an enzyme specific to each amino acid. The amino acid binds to the 3' end of the tRNA. ATP provides energy for this. The mRNA binds to the small subunit of the ribosome. Then initiation occurs where the ribosome reads 3 base codons at a time. The start codon is AUG. Then elongation occurs. A tRNA binds to the first binding site on the large subunit. It has a complementary 3 base anticodon. Secondly, another tRNA comes into the second binding site, and amino acids undergo condensation reaction forming a peptide bond. A third tRNA comes into the final binding site. And now the amino acids bond is acylated, which means the amino acids has formed the peptide bond and is ready to be detached. Translation occurs as the ribosome moves along the mRNA strand in a 5' to 3' direction, just like all the other processes shown in this clip. Then termination occurs when a 3 base sequence with no corresponding amino acid signal is detected by the tRNA. The final product is now a polypeptide, like for example hemoglobin, which is made up of 4 individual polypeptide chains.